Welcome to my channel, my dear friends. This is a concept for biomolecules, the enzymatic activity. Here we are going to touch each and every dimension of the enzyme activity which is present in the syllabus of class 11 and NEET. So let's start all together. The enzyme. According to the enzyme kinetics, this is the graph. The graph shows the enzyme kinetics rule. This is x-axis, this is y-axis. X-axis is for substrate concentration and y-axis is for velocity of the reaction. Now, as we add the substrate to the enzyme, the activity goes on, the speed goes on and on and on, and ultimately it comes to a constant value that is called Vmax, means velocity maximum. If we measure half the value of that is Vmax by 2, then it comes to, correspondingly it comes to Km. Km means the substrate concentration is Km, means a constant value called Michaelis Menten constant. According to the enzyme kinetics, E plus S means enzyme plus substrate is equal to ES complex, means enzyme substrate complex, that gives rise to E plus P. E means enzyme and P is product. Here we have K1, K2 and K3, which are the velocity constants for the respective reactions. So here we go, the equation obtained is the required velocity, that is small v, is equal to Vmax into S, means substrate concentration, by Km plus S, that is Michaelis Menten constant. So if V is equal to Vmax by 2, means our required velocity is equal to Vmax by 2, then Vmax by 2, we put the value over here. It appears like this. So we cross multiply and ultimately we arrive at this conclusion where Km is equal to 2S minus S, that is equal to S, where Km means Michaelis Menten constant. Now we are learning the classification of enzymes. The first one is oxidoreductase. The enzymes which are involved in oxidation and reduction reactions, means redox reactions, they are all known as oxidoreductase enzymes. Examples are all the dehydrogenase enzymes. All the oxidase enzymes and all the reductase enzymes fall into this category. Number 2 is transferase. These enzymes catalyze the transfer of functional groups from one substrate to another. That means phosphorylase, transaminase, transmethylase, they transfer the groups. So these are the examples. Coming to next part, that is third one, hydrolase. These enzymes catalyze the hydrolysis of various compounds. They are the digestive enzymes which we have in our digestive system. Example, trypsin, pepsin, amylase, lipase, carboxylase, carboxypeptidase, all of them are hydrolases. Next is lyase. Lyase are the enzymes specialized for addition or removal of water or ammonia or carbon dioxide, etc. from the substrate. For example, aldolase, fumarase, etc. Number 5 is isomerase. Isomerase are the enzymes that catalyze isomerization or epimerization reactions. For example, triose phosphate isomerase, etc. And ligases are those enzymes that catalyze the synthesis and binding reactions. For example, if you have known the DNA replication, there we have the ligase, DNA ligase, which joins the Okazaki fragments. I have already given you the video. Here the examples may be thiokinase, carboxylase, synthetase, etc. Now this is the basic structure of an enzyme and these are substrates. The substrates have different types of conformation. This is rectangular conformation, this is circular conformation, this is triangular conformation. and this is the active site where the substrate needs to bind. So specificity, it states that only this substrate will be able to fit over here. So this is called active site. Active site is the site where the substrate binds. The enzymes may have another site for binding the competitors, that is known as allosteric site. Enzyme activity can be regulated by either an activator or an inhibitor. The activator or inhibitor binds at a site other than the active site. That site is known as allosteric site. So this one. The enzymes which include the key metabolic regulators are called allosteric enzymes. So if any allosteric form is there, then that enzyme, that particular enzyme is called an allosteric enzyme. Now we know that there are two models given by two different scientists for enzyme action. One is lock and key model, which is given by Emil Fischer. Another is induced fit model given by Daniel Koshland. In lock and key model, the active site is almost static. In induced fit model, it is not static. In lock and key model, the active site of the enzyme precisely fits with the substrate, and here this does not completely fit with the substrate. There is no weakening of the substrate bonds in case of lock and key, so it is a rigid model. But in induced fit model, the catalytic group weakens the substrate bonds, so it is flexible kind of model. Now what is holo enzyme? Holo means complete. A holo enzyme or a complete enzyme is formed when the protein part called apoenzyme is attached to the non-protein prosthetic group called coenzyme. So this kind of videos will be provided to you on a regular basis. Please do subscribe my channel at the earliest, and please share the video to those people who cannot have the opportunity to hear these lessons like this. In lockdown period they are suffering so much. So please do it for their purpose. Thank you.